seminario de hoy del programa de Frontiers in Genomics, que organiza el Centro de Ciencias Genómicas de la UNAM y el Instituto de Biotecnología también de la UNAM. Entonces, el día de hoy tenemos al doctor Lluis Quintana Murci y la doctora Yvonne Rosenstein lo va a presentar en esta ocasión. Gracias.
Gracias, Mario. Pues bienvenido, Luis. Muchas gracias por haber aceptado la invitación que te hicimos ya hace varios meses. Nos costó trabajo encontrar una fecha y en medio de la pandemia era todo muy complicado. Pero bueno, ya lo logramos. Lo único que no logramos fue traerte a México. Pero bueno, esperamos que en alguna otra ocasión sea posible. Pues es para mí obviamente un grandísimo honor presentar al doctor Lluis Quintana Murci. Es muy difícil definir en 3 palabras y presentar en 3 palabras y rápidamente lo que hace en su laboratorio a través de una serie de metodologías que abarcan desde la genética de poblaciones, la genómica celular, el modelaje computacional, el trabajo con epidemiólogos, con genetistas, la antropología. Su investigación está sobre todo encaminada a entender cómo la selección natural, los eventos demográficos, el estilo de vida, todo eso ha influido sobre la diversidad del genoma de distintas poblaciones y cómo esto se traduce de alguna manera sobre la evolución de las enfermedades y la susceptibilidad a las enfermedades de las distintas poblaciones, ¿no? Entonces, sus áreas de estudio son realmente encaminadas a eso, pero se estudian muchísimos temas en su laboratorio y es, como les dije, muy difícil de definirlo exactamente. El doctor Quintana Murci, justamente estábamos platicando con Mario hace unos minutos, hizo su doctorado en la Universidad de Pavia, en donde, no lo sé exactamente, pero ya nos lo dirá, quizás tuvo la posibilidad o se formó directamente con el doctor Cavalli-Sforza, que fue realmente el promotor de toda esta parte relacionada con la definición de los rasgos genéticos de las distintas poblaciones. Entonces, realmente esto yo creo que lo ha de haber marcado en su formación. Y a partir de ahí es que después de su doctorado fue al Instituto Pasteur y de ahí pues no lo dejaron irse porque ahí sigue. Y ahora es profesor del Instituto Pasteur y desde el 2019 también ha recibido el nombramiento de profesor del Collège de France y ocupa la cátedra de genómica humana y evolución. Y pues sus pláticas son muy interesantes, son libres de escuchar para aquellos que las quieren escuchar, son muy buenas. Y ha recibido una enorme cantidad de premios y de reconocimientos a su excepcional trabajo y muy interesante. Y bueno, pues junto con esto decirles que tiene pues muchísimas publicaciones, más de 250 publicaciones de todo, en todos los ámbitos que él estudia, y realmente pues todas son publicaciones muy interesantes. Sin decir más, Luis, pues quisiera agradecerte el haber el haber aceptado participar en el curso de Frontiers in Genomics y en el ciclo de conferencias de Frontiers in Genomics.
Muchísimas gracias, Ivonne. Es un placer para mí estar con vosotros, aunque sea virtualmente. Y para responder a tu pregunta de si conocí o si me formé con Luca Cavalli-Sforza, la respuesta es sí, porque cuando yo hice mi tesis en Italia, entre el 94 y el 98, él pasaba 6 meses en el departamento, allí en Pavía, y 6 meses en Stanford, que es donde los últimos 30, 40 años hizo su carrera así. Y evidentemente, sí, me marcó mucho esa experiencia italiana porque fue allí donde yo descubrí la genética de poblaciones humanas y eso es lo que ha marcado un poco la continuación de mi trabajo. ¿Puedo continuar en inglés?
Claro que sí.
Okay.
Por supuesto.
Okay. Thank you very much, as I was saying before. And today I'm going to tell you about, and I hope to show you how we can use basically population genetic approaches, not only to understand, as we usually do for some students that were yesterday in my course, more kind of classic questions about population history, migrations, and admixture. But today I will kind of focus more on how we can use population genetics to tackle a number of questions more related to immunology and infectious diseases. So you know that in 2001, there was the sequence of the human genome, that it was very important for a number of questions in genomics, such as gene content, gene structure, etc. But in no way informed us about what we're interested in population genetics, that is diversity between individuals and between populations. And that's why there has been many different consortia in the last 20 years, like the 1000 Genomes, UK Biobank, and many others, whose main goal was to discover and to characterize diversity among individuals and populations from different regions around the world. So just for you to get an idea of how basically nonsense is to work with a single human genome reference sequence, is that if we compare the sequence of the genome of one of us, regardless of our ethnic or geographic origin, to the human genome reference sequence, each of us differ to the human genome reference sequence at least 10,000 amino acid altering mutations. Each one of us, we can have in between 300 and 400 stop, splice, or indels that can disrupt up to 300 genes. Each of us, we carry in between 50 and 100 mutations that have been already associated with a genetic disorder. The reason why most of us, we are healthy, is that we harbor these mutations at the heterozygote state. And also, we know today that depending on the demographic history of the population, the population will present a different burden of deleterious alleles. Meaning, for example, most, let's say, classical populations, like most Africans, most Europeans, most Asians, most Americans, were populations under demographic expansions, and we will show populations under demographic expansion show less of a burden of deleterious alleles, that populations that have gone through strong bottlenecks or founder effects or isolation, such as, for example, Sardinians or Finnish population or French Canadians. But the question is, what's the impact of all these mutations that we have in our genomes on phenotypic variation or even susceptibility to disease? So for that, to understand the impact of genotypes on phenotypes, there are these different ways to tackle this, and is using patients and using, for example, clinical genetic approaches where you are going to, for example, study a number of families that they have a particular disorder. And if you are lucky, you are going to find the gene that segregates with the disorder. And in most cases, these are rare variants that are associated with very severe disorders. In the other side of the spectrum, you have also genetic epidemiology approaches, or, for example, GWAS, by genome-wide association of studies, in which, for example, you are going to compare a few thousand of individuals that present severe dengue, for example, and you are going to match them by ethnic origin with healthy controls. And if you are lucky, you are going to find a number of variants that are common in the population, and they are not really causal, but just increase or decrease a bit your odds to develop the disease. But this is not at all what I'm going to talk today, because we don't work with patients. We work with healthy individuals, the general population, you and me, like the descendants of people that have survived across evolution, of Spanish flu to Black Death or tuberculosis. Don't forget that we are the descendants of those that have survived. So this kind of resilience is somehow engraved in our genes. And this is what we know how to do in population genetics. We know how to identify genes and variants that have been selected by natural selection with respect to others that have evolved neutrally. So why do we care about natural selection in the context of infectious diseases? First of all, because infectious diseases have been a major cause of mortality during human evolution. As you can see here in this survival curve, for example, Europeans in the end of the 19th century, which is basically yesterday from an evolutionary point of view, just 38% of Europeans reached the age of 40. You can see today here in red, the UK, 2000, that 98% of the population reached the age of 40. Of course, this massive increase in life expectancy is due to all what happened in the end of the 19th century or the beginning of the 20th century, the arrival of antibiotics, development of the first vaccines, and the improvements in hygiene. So, well, still, as you can see here, for example, Mozambique in purple, the same year of the UK, the 2000, only around 50% of the population reached the age of 40. So infectious diseases obviously keep on being a major cause of mortality. Who says mortality says natural selection. And this is what we are going to see. And another topic that we're quite interested in the lab is why we are different in terms of immune responses. Even in health, why some of us, we are here, that means that we have a very reactive immune response, which makes us more resistant to infectious diseases, but at the same time, the price to pay is that we are more susceptible to allergies or inflammation or autoimmunity. And why some of us, we are naturally here, that we have a kind of weaker immune response. So one of the themes of our lab, and you will see an example of that, is to try to understand the factors, genetic, epigenetic, lifestyle, many other factors that contribute to the naturally occurring variation in our immune responses. So today I'm going to tell you about 3 examples of research that we have been doing in the lab or that we are still doing. The first, I will tell you how we can use population genetics to understand genes that have played a major role in our survival against pathogens. Then I will give you a short but very precise and for me very illustrative example of how we can use ancient DNA to reconstruct an ancient epidemic. And I will give you the example of tuberculosis. And finally, I will finish with our more general population immunology studies or systems immunology studies, in which, as I've just told you, we try to measure the relative impact of the different factors that contribute to immune response variation in humans. So we start with the population genetics approaches in immunology. Basically, what we try to do is we study immune genes in order to see how they have been evolving during evolution and how selection has targeted these genes. You know that there is different types of selection. One type is purifying selection. In that case, this is the past and this is the present. In this case, you have a mutation in red, and this mutation is deleterious. Let's say that this mutation abolishes the signaling of a given immune pathway. So this mutation will be purged out of the population by purifying selection. So this mutation in the beginning, that's the frequency of the mutation, and that's the time. So this mutation in the beginning will evolve by genetic drift, but since it's deleterious, it will tend to decrease in frequency in the population. Then a mutation or a gene can be neutral. That means that even if there is this black mutation, so in that case, there is a dampening of the immune response through this receptor, it's not a big deal because there's another receptor that is doing the same thing. So in that case, seeing that the gene evolves under neutrality is basically telling us that the corresponding protein plays a redundant role in immune response. And finally, So in that case, the frequency of the mutation will just be governed by genetic drift over time. That means random processes. And finally, we have a positive Darwinian selection, in which you have this mutation in green, for example, and this mutation makes that you are being more efficient, for example, in a given immune response. So this mutation will obviously increase in frequency in the population because, for example, let's say it protects against malaria. So over time, this mutation will tend to increase in frequency in the population by positive selection. So over the last 10 years, we have been studying many families of microbial sensors, or innate immunity receptors. These are genes that encode for receptors that basically they are recognizing that there are virus or bacteria that have to get into your body. So basically they are the first line of host defense against pathogens. And depending on how selection or neutrality has acted on these genes, we have been able to make a hierarchy of those that are essential and non-redundant, in red here, to those that are redundant, in gray, for example, depending on how selection has acted on them. And for example, we have shown that endosomal toll-like receptors that are specialized in the sensing of nucleic acids, particularly from viruses, evolve under strong purifying selection, which means that they are essential and non-redundant, with respect to others like toll-like receptor 5 that present higher levels of immunological redundancy. For example, toll-like receptor 5 present a mutation, a stop codon mutation, that is present in between 10% and 30% of populations in Europe and the Mediterranean basin. And this mutation acts in a dominant negative manner. This means that up to 30% of individuals can be knocked out for TLR5. So this means that just TLR5, the function fulfilled by TLR5 is redundant. In this case, is the sensing of flagellin, of flagellated bacteria. And when you don't have TLR5, it's not a big deal because you have other receptors that will take over the same function. But also, we have found signatures of positive selection. This means that we have identified particular mutations in particular groups of genes that have evolved, that have conferred specific selective advantage to specific populations. This is the case, for example, of these mutations in type 3 interferons that we identify as being under strong positive selection in European and East Asians. And proof of concept that we're identifying advantageous mutations is that some of them have been also associated with a spontaneous viral clearance of hepatitis C virus. This is another group of mutations in the cluster TLR1, 6, and 10 that we found to be under strong positive selection in Europeans, and this cluster is associated with a reduced inflammatory response. And I will come in a minute to this. But then the next question to address over the years was, let's take all the mutations in innate immunity genes that have been under strong positive selection, and let's date the time at which selection started to occur, because in population genetics, we can do that. And basically, in most cases, the time of selection, of positive selection, was in between 3,000 and 13,000 years ago, which is the period in which populations started to shift around the world from being hunter-gatherers and nomadic people to being sedentary farmers. But something that I wanted to tell you more about today was how admixture can be a source of advantageous variance. We know that modern humans left Africa around 60,000 years ago. And when they left Africa, they found in Eurasia Neanderthals, in Western Eurasia, and Denisovans in Eastern Eurasia, with whom they had mixed. You know that all of us today of non-African origin, we have in between 2 and 2.5% of Neanderthal ancestry in our genomes. So the idea, the question we wanted to address was the extent to which this Neanderthal ancestry that there is in our genomes, have they been beneficial for us, or this introgress material was evolving just neutrally? And we wanted to address that in the context of immunity, because it's one of our main interests. And in this first study, what we did, the question was, let's see if innate immunity genes are enriched in Neanderthal ancestry with respect to the remainder of the genome. Because if the answer is that they are enriched, that means that when early Europeans, early Africans in that case, came to Europe, they had to adapt to a new environment, climatic environment, but also new pathogens. But Neanderthals were there for 300,000 years. So the idea was that if innate immunity genes are enriched in Neanderthal ancestry, this means that this admixture of early Europeans and Neanderthals contributed to Europeans advantageous mutations that allowed early Europeans to better survive the pathogens they were encountering in Europe. So for that, we measured the levels of Neanderthal ancestry in around 1,500 innate immunity genes. And actually the answer is yes. Both in Europeans and in Asians, innate immunity genes are enriched in Neanderthal ancestry with respect to the remainder of the genome. Here you have an example of the TLR1-610 cluster. These are around 100 individuals, and each line is an individual. And basically, when you have a white dashed line, means that the individual has a kind of Homo sapiens haplotype. These are present-day individuals of European origin, like us, right? So, and when there is a blue line, means that this individual, the region is completely covered by a Neanderthal haplotype. And you can see that around 35–37% of Europeans have the whole TLR1-610 region completely covered by a Neanderthal haplotype. At the functional level now, we're trying to understand which is the evolutionary advantage of having this Neanderthal haplotype in the TLR1-610 region. And also we have found other genes that you can see here, some examples that are part of the innate immunity system that present among the highest levels of Neanderthal ancestry of the human genome. Another thing that we have observed in monocytes that are a major cell type of innate immunity cells, that what we have found is that Eqtls, that are expression quantitative trait loci, in other words, are mutations that are associated with changes in gene expression, are enriched in Neanderthal ancestry in monocytes, but particular mutations that are associated with changes in gene expression related to antiviral responses. In this case, is upon activation of the TLR7/8 pathway or infection of monocytes with a live strain of influenza virus, which suggests that Neanderthal introgression was particularly beneficial for allowing early Europeans to better resist viruses in particular. We have been also interested in trying to understand how Neanderthal introgression has affected other layers of gene regulation, such as promoters, enhancers, or microRNA-mediated regulation. And basically what we have seen is that the only regulatory elements that as a whole are genuinely enriched in Neanderthal ancestry are enhancers that are active in fat tissue or in T cells. And as you can see, these enhancers enriched in Neanderthal ancestry, they are enriched so in all types of T cells, but not in other cells like NK, B cells, or monocytes. More recently, also, we turn our attention to Pacific populations. And why Pacific populations? Because they are supposed to have the highest levels of combined Neanderthal and Denisovan ancestry worldwide, because they have this 2.5% of Neanderthal ancestry that all non-Africans have, plus the Denisovan ancestry that they inherit from their Asian ancestors. So for that, we analyzed around 300 whole genome sequences from near and remote Oceania. And what we observed is that while Neanderthal ancestry is very homogeneous across near and remote Oceania, from 2.2 to 2.9%, Denisovan ancestry is very heterogeneously distributed, going from almost 0% in Taiwan or some populations of the Philippines to 3.2% in Papua New Guinea and some populations of the Vanuatu Islands. But the next question was, which phenotypes this Neanderthal introgression or Denisovan introgression are mostly affecting in modern humans? And the surprise was that while Neanderthal ancestry is associated with multiple phenotypes related to immunity, neuronal development, metabolism, and pigmentation phenotypes, the contribution of Denisovans is almost exclusively related to the regulation of innate and adaptive immunity, suggesting that Denisovan introgression was particularly important to resist, was particularly beneficial for modern Pacific Islanders to resist the local pathogens they encountered. So let's go to this example of how to use ancient DNA to reconstruct an ancient epidemics. And for that, let's focus on tuberculosis. Tuberculosis, as you know, is one of the 3 diseases that are the deadliest at the worldwide level, together with HIV and malaria. And despite in Europe, as you can see in this map, we are doing relatively well today. Tuberculosis is considered to have been the deadliest infectious disease during European history. But in terms of the host genetic basis of tuberculosis, we know very little. There were some examples, of course, of mutations restricted to some particular families that were associated with tuberculosis-like phenotypes, such as Mendelian susceptibility to mycobacterial disease, and all the fabulous work that Jean-Laurent Casanova is doing at Rockefeller University in New York. But more recently, for the first time, this laboratory at Rockefeller identified the first common mutation in the population. Is present in between 2 and 4% of the population. Watch out, this is not 25%. This pie chart is 2.5%. So this mutation is present in between 2 and 4% in populations of Europe, or in general, populations of European ancestry, like in North and Central and South America. And this mutation that is located in this gene, TYK2, is associated with a higher risk to develop clinical forms of tuberculosis. So it may seem low frequency, but don't forget that 4% of the populations is millions of people that have this mutation. So in that case, our idea was, okay, so let's try to study the history of this mutation to try to better understand the history of tuberculosis. And for that, what we did is we analyzed 1,000 genomes of ancient people that have lived in Europe over the last 10,000 years. From the Mesolithic period, around 10,000 years ago, until today. So through the early late Neolithic, the Bronze Age, the Iron Age, the Middle Ages, and today, right? So what we observe is that the mutation appears from the first time in the Mesolithic, in Anatolia, what's today present Turkey, and that the mutation increased in frequency over time, as you can see in these curves, to reach almost 10% at the end of the Bronze Age. And then from the Bronze Age on, it dramatically decreases in frequency until the 2-4% of today. So in population genetics, we know how to distinguish if these changes in frequency are due to random processes like migrations, like drift, or if they are due to selection, in particular negative selection, For example, is this decrease in frequency due to negative selection, which would mean that around this time, tuberculosis appeared, and that's why the mutation started to be purged out of the population? So this is what we did using approximate Bayesian computation. And the first thing we observe is that the risk mutation appeared around 30,000 years ago in the ancestors of Western Eurasians. But more interestingly, what we show is that this mutation has been evolving by genetic drift, and just over the last 2,000 years, this mutation has evolved under a strong negative selection in Europeans with a selective coefficient that is one of the strongest of the human genome, what supports the notion that tuberculosis has imposed a heavy burden on European health over the last 2 millennia. So now we have seen basically where do we come from, how and with whom we have admixed, how admixture with Neanderthals, for example, or Denisovan has conferred selective advantage to modern humans for some particular functions, including immunity. But here I want to share with you our more recent interest about to try to better understand which are the factors that make us different with respect to immune responses. Why some of us were at higher risk to develop a severe COVID, and why others are more resistant. So for that, we use systems immunology approach in which we work with different individuals from different populations, and we can work either with whole blood that we infect with pathogens or purify cell types. And then we measure gene expression before and after immune activation or infection. And then we map the genetic basis of changes in gene expression related to immune responses. And this is what's called Eqtl mapping, the mapping of expression quantitative loci. Just in case you are not familiar with that, this, for example, this is a SNP, and you can see that regardless of the genotype does not affect the levels of gene expression. So this SNP is not an Eqtl. But this SNP, SNP2, is an Eqtl, because you can see that depending on your genotype, you are going to express more the gene. But we're particularly interested in this kind of Eqtls that are called response Eqtls. That is, the SNP3, if you don't stress the cells, has no impact on gene expression. But if you infect the cells with MTB or flu, the mutation is going to have an impact on gene expression. This is called a response Eqtl, and this basically mirrors gene-environment interactions. So we have been working with 2 cohorts to understand which are the factors that participate in immune response variation. And the first one is Evo-Immunopop. So Evo-Immunopop is 2 populations, both living in Belgium, one of African ancestry and the other of European ancestry. And we've extracted from these populations monocytes. So again, a major cell type of innate immunity. And monocytes were exposed to different TLR ligands. Basically, so there are ligands that activate major pathways of innate immunity, right? But also they were exposed to a live strain of influenza virus. So, and then we did RNA sequencing and Eqtl mapping. So first thing we observe in this PCA is that, as you can see, this is a PCA based on gene expression, that what explains most of the variance is the condition of stimulation. Non-stimulated cells, influenza virus, and the different TLR pathways. But if you zoom into one of the conditions, you can start to see that there are subtle differences in the way Europeans and Africans respond to infection. And to make a very long story short, what we found, obviously, is that there is no on-off differences in between the two populations. But what we found was marked differences in the intensity to which they respond to immune activation or infection. And more importantly, what we found is that 80% of the difference of the population differences in immune responses can be just explained by EQTLs. That means mutations that control gene expression in the context of immune responses, and that these mutations are just differently distributed in terms of frequencies between Europeans and Africans. Sometimes a single mutation, like this one, TLR1, a mutation in this gene that is associated with a drastic reduction in NF-kappa B activity, and this mutation is present in 70% of Europeans and 2% of Africans. And this mutation is a master regulator because this mutation is associated with a decreased expression of around 100 pro-inflammatory genes. In other words, the 70% of people of European ancestry that have this mutation, we have a reduced inflammatory response. And this mutation is the same that I told you before, that is under a strong positive selection in Europeans, showing that a slightly reduced inflammatory response has conferred a selective advantage. So kind of highlighting the general balance that the innate immunity system has to reach. That is, well sense the pathogen, well recognize the pathogen, switching on an inflammatory response, but not super strong, because if not, the inflammatory response, of course, will kill the pathogen, but will kill also the host. The second cohort is called milieu intérieur. This means internal milieu. And in this case, we wanted to take a bit step back and maybe reduce genetic diversity. So not compare different populations, just study a single population, but with much more parameters. So in order to try to understand other phenotypes, other factors that may participate in immune response variation. So for that, we recruit 1,000 individuals in Brittany, in Western France, and stratified by age and sex. So 5 decades of life, from 20 to 70 years old, half males, half females. From each of them, we have different materials, whole blood, serologies, nasal swabs and stools, skin biopsies, and very important, a 44-page questionnaire about sociodemographic variables, vaccination, medical history, nutrition, physiological measures, etc. And the whole blood of each individual was ex vivo stimulated with 30 different ligands, going from real microbes, viruses, bacteria, or fungi, to activators of the innate or the adaptive immune system. So let me share with you just 3 examples of this consortium. In the first, the question we wanted to address was, which are the factors that participate in the natural heterogeneity that there is in our blood of different cell types? What makes that some of us will have more CD4 T cells or B cells or monocytes or whatever? So for that, we took the 1,000 individuals and we defined more than 70 different subpopulations of cells in our blood. And then basically the question is, which of the 140 variables that we have for each individual, age, sex, genetics, nutrition, physiological measures, past vaccination, how all these variables affect cellular heterogeneity in whole blood. And basically we found that out of the 140 variables, just 5 have a real impact on immune cell heterogeneity, and it's host genetics, CMV seropositivity. This, you know that many of us were chronically infected with cytomegalovirus, the age of the individuals, the smoking status of the individuals, and the sex of the individuals. And basically what we found, which makes a lot of sense, is that while genetics, in blue here, explain most of the variation of innate cells, non-genetic factors explain a large substantial part of the variation of adaptive cells. In the second study, we did the same thing. This was in collaboration with Genentech, and the question was the same, was which factors affect the variation of our gut microbiota? So for that, of the 1,000 individuals, we take their stools and we did shotgun sequencing. And basically what we found is that 15% of the species of our gut microbiota are differentially abundant depending on sex, and 40% of the species changes across age. And of course, also, we found, apart from sex and age, as you can see here, I'm not getting into the details, we found other factors that also affect gut microbiota, like nutrition and other lifestyle variables. But all of that was at the steady state, both the cellular heterogeneity or the microbiota is without stressing our immune system. So we wanted to understand which are the factors that participate to the immune response variation when you stress the cells. So for that, in the 1,000 individuals also, we assess the transcriptome following whole blood stimulation with Escherichia coli, Staphylococcus aureus, BCG, that you know that is what we use as a vaccine for tuberculosis, a fungus, Candida albicans, and influenza as a virus. And here you can see again the principal component analysis. Here you have the non-stimulated individuals, here the influenza virus and the others. And you can see already that there is quite variation even within a single condition like influenza virus, for example. And when we try to pinpoint which are the factors that participate to this variation, we found that most of the variance is explained by most of the variance in immune responses to infection is due to our natural variation of different immune cell types in our blood, followed by genetic factors. Oh, sorry, I have a problem with my slide. Okay, now, followed by genetic factors acting both in cis and in trans, followed by age and sex. Of course, these are average across all genes. Some genes, most of the variation is due to genetics, or most of the variation is due to sex. But still, a large fraction of the variance in immune response variation still remains unexplained. So our perspectives in terms of population genetics, we keep on working on population history in Polynesians, and we try to make some connections between genetics and metabolic diseases in Polynesia, such as diabetes and hypertension and obesity. We keep on working a lot with how archaic admixture or modern admixture has participated to the adaptation of our species to different environments. And in terms of populations and systems immunology, we keep on working on how rare variants in our genomes or epistases, that mean combinations, participate to immune response variation, how age and sex also interacts with the genetic or epigenetic control of immune response variation. And also we work now at the single cell level using influenza infection as a model. And if you still allowed me 3 minutes, I have a little bonus of our most recent results that is very preliminary, but I want to share it with you about what's unique of the immune response to SARS-CoV-2 with respect to other RNA viruses, such as influenza virus. So these are the type of questions we want to address. Many of them we have not addressed yet because we started, but we want to understand why different populations like African, Europeans, East Asians, but also we are starting a collaboration with Macarena Fuentes in Chile to also understand also some populations of Native Americans. And I would be most than happy if some of you want to participate also in this project of how ancestry and how admixture affect immune responses to SARS-CoV-2, because as you know much better than me, in Mexico you have a huge amount of diversity. Basically, just in one, like Brazil or like many other countries, you recapitulate the variation of the whole world, basically. So just let me share with you our preliminary results of that. So what we do is we take PBMCs, so from blood of that we expose PBMC to SARS-CoV-2, and as a control or as a comparison also to influenza A virus, and we do single cell sequencing. So as you can see here in our preliminary results, we did a kinetic study just based on 8 donors that we infected them with both influenza and SARS-CoV-2, and at 2 time points, T6 and T24 hours. As you can see here, this is T0, this is T6 hours, and this is 24 hours. And the color codes are like here, non-infected in black. In kind of gold, you have SARS-CoV-2 cells, and in kind of dark brown, you have influenza virus. So in this plot, you can see that most of the variation is just due to being exposed to the virus or not being exposed to the virus, regardless of which virus we are talking about. With some exceptions, by the way, here, of some European individuals that were infected with SARS-CoV-2 and behave as if they were non-infected. And the second factor that explains most of the variation in gene expression at the single cell level is the time point, 6 hours versus 24 hours. But when we go specifically to the number of genes that are differentially expressed in different cell types, as you can see here, here you have it in blue, SARS-CoV-2, and in red, influenza virus. So basically, to make a very long story short, as you can see, in most cases, the changes in gene expression are highly correlated. You have on the x-axis the SARS-CoV-2 response, and on the y-axis the influenza response. So the two responses are highly correlated. There is a tendency of more genes being dysregulated by influenza than by SARS-CoV-2. But the cell type that presents the least correlation is the myeloid cells. And for example, talking about them here in myeloid cells, here you have again the SARS-CoV-2 response versus the influenza response. And in gold, you have the genes that seems to be specifically dysregulated upon SARS-CoV-2. And we have identified around 400 of these genes that are more strongly upregulated upon SARS-CoV-2 challenge with respect to influenza. And here you have a number of examples. And basically these genes that appear to be more specific to the SARS-CoV-2 response are enriched in genes that are part of the inflammatory response. And in terms of population differences, I cannot tell you too much because with 8 individuals for the moment is very few, but now we have finished 300 individuals, but for the moment we have identified 17 genes whose expression differs in a consistent way between African and European cells across cell types, time points, and conditions. And here you have 2 examples like EL32 and Eqtl M3, that is a non-restriction factor. So with that, I would like to thank my lab, my collaborators, the funding agencies, and of course, you for your attention. And I'm very happy to take questions. Thank you.
Thank you very much. Go ahead.
Thank you so much for this wonderful talk. And I don't know, we will wait. I mean, if people want to make questions, you can put them through the chat. We will read them. And those who are on YouTube, you can also put your questions and they will some way be brought to us. No sé si haya preguntas.
Espero no haberos aburrido con tanta información, ¿eh?
Pero
No,
para nada.
Para nada, al contrario.
Estuvo muy, muy interesante.
Yo tengo una pregunta, no sé en qué idioma formularla.
En español.
La voy a formular en español. Me interesa mucho lo de los enhancers de ascendencia neandertal. De alguna manera estamos haciendo algunas cosas con enhancers. Me pregunto si han analizado en más detalle qué motivos son diferentes que están presentes en estos enhancers neandertales comparados, por ejemplo, con la población africana.
No lo hemos estudiado.
¿Qué factores transcripcionales podrían estar ahí involucrados? Porque creo que sería muy interesante. Y también
es muy interesante. No, pero tenemos que hacer un follow-up de esto, o alguien tiene que hacer un follow-up, porque por el momento no lo seguimos, porque además coincidió que fue cuando empezó todo lo del COVID y prácticamente después en Pasteur solo nos dejaban trabajar si trabajamos en COVID, y si no, todos a casa. Por eso empezamos a trabajar un poco en COVID.
Todos empezaron a trabajar en COVID. Bueno, era una manera de obligarlos.
Veo que hay algunas preguntas.
Sí. En el chat hay algunas preguntas y hay algunas manos levantadas también.
Ok, veo. Concerning your research, puedo leerla si queréis. In monocytes and innate response. Es de Edgar Leal. So, concerning your research in monocyte and innate response with response Eqtls, would you expect similar results with adaptive response? I would find interesting the evolution within exhausted and Treg cells. Yeah, that's super interesting. Unfortunately, probably because of our ignorance in adaptive immunity and the genomic difficulties associated with some parts of the adaptive immunity, like HLA, T cell receptors, B cell receptors, that are very complex regions, sometimes we just don't look at it. So I think this is a fabulous line of research that has to be done, but we haven't. So there's another question from Arnaud Ronser. Ronser. Okay, I didn't want to pronounce it in the French way. Okay, Arnaud Ronser. Great talk. Do you observe associated recombination hotspot near pathogen-resistant gene in human genome as it is in plants? Pathogen-resistant gene. Never look at that. It's very difficult also to define what's a pathogen-resistant gene because there is so many. But that's true that what we could do is to take, for example, because we have very well annotated them, all innate immunity genes, and to look if with respect to the remainder of the genome, there is more hotspots nearby. That's a wonderful suggestion. Thank you. Esperanza Martínez, ¿por qué se propagan mutaciones como las de TIC2 si hacen a las personas más sensibles a tuberculosis? Hace muchos años se decía que mutaciones deleterias podrían perder algún beneficio. Ay, se ha ido esto. ¿Podrían tener algún beneficio en otra vía? Hombre, la respuesta ahí es ¿por qué se propagan? Nuestra hipótesis en el caso preciso de TIC2 es que se propagó porque hasta ese momento la tuberculosis no era suficientemente endémica o pandémica para que hubiese una fuerte selección negativa. Entonces la mutación se simplemente toleraba porque en ausencia de tuberculosis o muy poca sobrevivía. Cuando la tuberculosis empezó a estar muy extendida hace 2000 años, fue en ese momento que empezó a estar bajo Purifying Selection. Pero hay muchos otros ejemplos, como dice esta señora, de mutaciones que han aumentado en frecuencia en la población, que hoy son deleterias y que aumentaron en frecuencia en la población simplemente porque estaban asociadas a resistencia contra enfermedades infecciosas, pero después en ausencia de las enfermedades infecciosas, estas mismas mutaciones están asociadas hoy a increased risk of allergy, inflammation, and autoimmunity. That's all what's the hygiene theory. And these very nice studies, in great part epidemiological studies, that show, for example, that at least in Europe, when there is a decrease of infectious diseases, started in the 1950s, 1960s, is at the same time that there is a massive increase of autoimmunity, inflammation, and allergy. Hay otra pregunta.
Hay varias.
Could we find these same variations in gene expression between populations in all immune cell types? Or are these variations specific to monocytes, macrophages, and so on? That's a very good question. The answer is yes and no. Many Eqtls are cell type specific, but some Eqtls are a bit universal. So the answer is you are right, but many Eqtls are really cell type specific. Hi, are you interested in comparing the response between different types of coronavirus like MERS? Do you think the relevant genes will be different between them? I don't know. I have no idea. That depends a lot on the tropism of the virus. There's so many things. I'm not a virologist, not an immunologist. So sorry if my questions are a bit evasivas, because sometimes I just don't know the answer. But of course, this is very interesting. Marta Vázquez, have you seen linkage between mutated genes and mutations in genes encoding proteins acting together with the protein originally detected for mutations? Or for example, have you seen linked mutations in different genes encoding proteins acting in the same enhancer? We haven't looked at that, but that's very difficult. That's very interesting. That's biologically, that makes a lot of sense. And I think we are massively missing a lot of the genetic basis of immune response variants, either because we just focus on common variants, but I think a great part of the missing heritability that is called is due to combinations of rare variants or combinations of rare variants with common variants. But still, there is a huge amount of work to be done in terms of methodological developments to detect epistasis and all this. Is there a link to reduce inflammation responses by TLR mutation and reduce cancer? I don't know. Sorry for that.
There is one hand, Edmundo Calva. Maybe we can give him a chance to talk.
Hola.
Hola.
Mi pregunta es un poco paralela a este gran estudio que nos presentaste. ¿Sabes si hay algún tipo de estudio similar, pero con otras características humanas, por ejemplo, el desarrollo del sistema nervioso o algunas otras características humanas que en los últimos, digamos, 10,000, 100,000 años nos han dado una ventaja similar a lo que nos has presentado en el sistema inmune?
Sí, hay, de hecho, esta mañana estaba hablando con alguien en el Weizmann Institut en Israel que trabaja sobre la morfología, es decir, cuáles son los genes que han participado prácticamente en la hominización de nuestra especie. Y lo que compara y hace un trabajo, bueno, que a mí me ha parecido espectacular. Lo que hace son células híbridas entre humanos y chimpancés. Yo no sabía que la tetraploidía, que las células sobrevivían y las podías después mantener en cultivo celular, y él lo hace, y lo que hace es perfecto, porque él lo que quiere comparar es cuáles son los genes y las mutaciones que han participado en nuestra formación esquelética, morfológica, and eventually quiere hacerlo a nivel de brain, also, del cerebro. Y lo que hace, como evidentemente somos dos especies distintas, con tantos environments distintos, lo que hace ahí es un sistema súper controlado, porque en células híbridas es el mismo cellular environment con los mismos transcription factors and trans thing, y lo único que va a detectar de diferente es lo que actúa en FIS. Me parece espectacular. Para responderle a la pues
excelente,
sí,
muy interesante. ¿Quién es el autor, perdón?
David Gockman. Se escribe G-O-K-M-A-N. Es un joven brillantísimo que hizo su tesis en el Hebrew University en Jerusalén. Después hizo su postdoc en Stanford y ahora ha montado su propio grupo en el Weizmann Institute en Rehovot.
Excelente, muchas gracias y felicidades.
Gracias. ¿Hay alguna otra pregunta? Ah, ya, veo otra. Have you found in the polarizations of cell types also differ between populations, as polarization between M1 or M2 microphages or T1? We haven't gone so far for the moment. Es difícil porque normalmente tener células de poblaciones diferentes no es tan fácil. Pero bueno, una cosa que si uno quiere estudiar bien diferencias entre poblaciones que son debidas a la genética, creo que ya lo hablaba ayer con uno de vuestros estudiantes, creo que las poblaciones mezcladas, admixed, en Latinoamérica son un sistema perfecto porque tenéis en la misma población varias ancestrías. Entonces, que por definición están expuestas al mismo environment, porque es el mismo individuo, right? Entonces, os quiero highly encourage, si queréis trabajar de esto o que trabajemos juntos, porque me parece súper prometedor. Y sobre todo lo que me parece prometedor es que, lo que decía ayer también a los estudiantes, que yo ya estoy harto de trabajar con European Ancestry People, porque 78% de los estudios genómicos se hacen en europeos. Y los europeos solo son el 16% del planeta. Entonces, creo que estos estudios se tienen que hacer en otras regiones también.
Sí. Totalmente de acuerdo.
Espero que haya colaboradores.
Ojalá.
De esta parte del planeta.
Ojalá.
Muchas gracias por la invitación y que sepáis que estoy dispuesto a volver a haceros un talk cuando queráis y que deciros que entre ayer y hoy my day end very nicely porque es súper, ¿cómo se dice? Lo contrario de frustrante, ¿cómo se dice? Se me ha olvidado. Estimulante.
Okay. No, pues muchas gracias a ti. Realmente fue un honor que participaras en el programa. Estamos muy contentos y ojalá sí volvamos a tener la oportunidad de que nos vuelvas a dar una plática y clase en el programa, pero que ya sea aquí en México.
Ojalá.
Sería extraordinario.
Un placer.
Sería fabuloso. Pues muchísimas gracias de nuevo y pues todo muy interesante realmente y muy sugestivo, ¿no? De otras cosas.
Exacto.
Muchas gracias.
Yo voy a buscar a los enhancers de Neandertal, a ver qué hay ahí.
A ver.
Si encuentro algo te digo.
Muy bien, pues muchas gracias entonces.
Hasta pronto, que tengáis un buen día.
Gracias, perfecto.
Hasta luego, muchas gracias.
Gracias.
Hasta luego.
Hasta luego.
Gracias, hasta luego. Gracias a todos por asistir al seminario.